So in this picture you see Thomas Hunt Morgan, who worked in the early 1900s. He was a fly geneticist who first figured out and figured out that there was something that he eventually called linkage. Linkage means, it can mean the location of a gene on a chromosome, so you can say the Huntington's disease gene is on chromosome 4, so it's linked to chromosome 4. And you can also say that two genes that are close together on the same chromosome are linked to each other. So two genes close together on the same chromosome are linked, and they're also linked to that chromosome. But Thomas Hunt Morgan, I really don't believe he set out to find linkage. I mean, how could he? He didn't know it existed. But what he was trying to do really was figure out the inheritance pattern of a mutant fruit fly trait, white eyes. And so he found a fly that had white eyes. Red eyes are the normal allele, the most common allele. We call it wild type. And so he had a fly that had white eyes, and he wanted to know if it was dominant or recessive. So he set up what's basically a Mendelian Cross. You can see in this picture he started with a red-eyed female crossed to a white-eyed male. And the hybrids all had red eyes. So that really answered one of the questions. It's a dominant, the red is dominant to the white allele. But then when he mated brothers to sisters, the hybrids, he got an unusual result. Now, it's not unusual in that you do have 3/4 red-eyed offspring for every 1/4, or for every one white-eyed offspring, 3-to-1 ratio. That's perfectly Mendelian. But what he noticed as he was actually sitting and counting these flies and writing down which ones had red eyes and which ones had white, he noticed that all the white-eyed flies were all males. I mean, you would expect half would be male, half would be female. So that's an unusual result. Why were they all male? Okay, so the phenotype ratio with regard to eye color was normal, but the ratio with regard to gender, sex, was not. So that cued him in that there's something weird going on here. Now, in this image, you have the genotypes given to you, but he wouldn't have known that because this notation that they're using already tells you that this gene is on the X chromosome. Because when a gene is linked to the X chromosome, you actually put the allele, the little symbol here, as if it's a superscript on the X chromosome. So that means linked to X. And that you only use this notation when you know that a gene is linked on X. So anyway, he got this result, this unusual result, with all the white-eyed flies being male. So he set up what's called the reciprocal cross. Now the female has the white eyes and the male has the red eyes. Anytime Mendel did a reciprocal cross, it never affected the results at all. He always got consistent results. But this time, when Morgan did the reciprocal cross, right away in the F1 generation, he sees something different. In the F1 generation, half the flies have red eyes and half have white, which is weird. And then when he crossed brothers to sisters, he gets in the F2 generation, half the flies have red, half have white. But what is kind of normal in this case is half the red-eyed flies are male and half are female. Half the white-eyed flies are male and half are female. So you get kind of these results that you wouldn't normally see, except the only way to explain it is if this gene for eye color is linked to the X chromosome. It's carried on the X chromosome, so not only when that allele is being passed to a gamete, it's also traveling with a chromosome that has something to do with the sex of the next offspring. So this was Morgan's work, and I would say Morgan's unusual result was that when he did what he thought was a Mendelian cross, even though he got a 3-to-1 ratio of offspring, all the recessives were also male. Now, how does this kind of translate or transfer to like what happens in humans? In humans, it's very typical for an individual who has a certain X-linked disorder to be male. And the reason is that alleles, disease alleles that are recessive are rare. And for any rare allele, I mean, by definition, the chance of getting it is rare. And so a male only has to inherit one rare allele on his X in order to show that phenotype. And that's because the Y chromosome doesn't have any of the same genes. So whatever gene the male gets, whatever allele of that gene he gets on his 1X chromosome from his mother, that's the trait he's going to show. A female, on the other hand, she would have to inherit not just one rare allele, but she would have to inherit 2 rare alleles. And so inheriting one rare allele, like a male, is easier or is more likely than inheriting 2 rare alleles. So that's why we say females with recessive X-linked disorders are much rarer, more rare, than males with X-linked recessive disorders. So how do you identify sex linkage in a pedigree? It's actually not easy to do in a single pedigree. Remember that 22 out of the 23 chromosomes are not sex chromosomes. So in general, that's, the most likely for any gene is that it's not on the X. If you were just randomly picking, which side would you be on? You should always think that a gene is autosomal unless there is some information in the pedigree that forces you to even question that. And what do I mean by information in a pedigree that forces you to question that? I mean, if you see affected individuals are always squares, which are males, if the shaded ones are all squares, then that will force you to at least consider that this gene could be on the X chromosome. Remember that if a mother is a carrier, if she's heterozygous, then each of her sons has a 50% probability of being affected by the rare phenotype. Each of the daughters only has a 50% probability of being just a carrier. So if the mother's a carrier, the daughters are safe from having the disorder. The sons are at a 50-50 chance of having the disorder. Again, the risk for a boy is higher. It's more likely to have a boy with the disorder than a girl. Right. And so there's some logic to it. If a female is affected, she must have gotten one of her alleles from her father, and he must then be affected because he only has one X. The sex-linked genes in humans that are pretty well studied are hemophilia. There's actually a couple genes for hemophilia, which are clotting factors. The ability to clot your blood has to do with clotting factors, and if you have a defective gene for those, then you won't be able to clot the blood. Colorblindness, which colorblindness isn't so serious problem, but it is on the X chromosome. And Duchenne muscular dystrophy, which is a very severe disorder. So hemophilia and Duchenne muscular dystrophy, both life-threatening, and then colorblindness, not really that serious. All right, now, here's an example of a pedigree where I would allow you to entertain the possibility of sex linkage. So like I said, if you see circles and squares that are shaded in, you need to default always to autosomal in terms of predicting the mode of transmission. But if you see something like this, then I would allow you to consider sex linkage. So all the shaded individuals are squares. Not all the squares are shaded, that's different. But all the shaded individuals are squares.